Hello biology apprentices, today we're going to talk about single trait crosses using the Punnett square. So let's go to it. First, we're going to make a review about some of the most important genetic concepts that you need for answering these problems. Let's remember what is genotype. The first three letters give you the hint. Genotype will be the genes or the genetic composition of an organism, the actual types of alleles that this organism has. Phenotype will be the physical traits or the physical properties, any measurable or physical feature that we can observe in an organism. Homozygous, when the genetic composition of an organism has two alleles of the same strain or the same type. It could be dominant homozygous, like the example we are seeing here, or recessive heterozygous. Hetero means different. In this case, one dominant and one recessive. Dominant gene or dominant allele, it will be the one that masks gene. Recessive gene, a weak gene, a one that needs the presence of another recessive to produce a certain phenotype. Remember that dominant genes will be represented with uppercase letter and lowercase will be used in the case of recessive genes. Now, the exceptions of these rules. Codominant condition or codominance. If we have two traits, in this case red and white, a codominant condition will produce spotted phenotypes in the offspring. It means two phenotypes at the same time. Incomplete dominance, when if we have a red and white phenotypes in the parents, the offspring will have an intermediate version of these two parental phenotypes. In this case, a pink phenotype. So, once you have these concepts clear, let's go to the problems. My suggestion for the problems is that you pause the video, you write the problems in your notebook, and then you answer them by yourself once we have finished the explanation. So, the first exercise goes like this. In cattle, the red 4 is dominant over the white 4, which is recessive. And we have here two letters to represent these genes. A uppercase for red, A lowercase for white. The question says, if a cross is made between one red homozygous and one white homozygous cattle, calculate the ratios of phenotype and genotype of the first generation. This is how we represent the first generation or the offspring, F1, filial 1 or first generation. Where do we start? As any problem, you need to take out the information that comes within the problem. The problem is giving you the phenotype of the parents. Here you have the colors. Remember that phenotype are the physical traits. And you have also the genotype, the internal condition of the genes of these parents. One parent will be red 4, another parent will have white 4, which is the other phenotype, and the problem clearly established the genotypes. The red one is homozygous. Now remember that homo means same, so the genetic composition of the red 4 organism will be two uppercase letters. Now the other organism, the white one, it is also homozygous. So in this case, it will be two lowercase letters. Once you have established the gametes for the parents, you have done half of the work. The rest is to fill up the Punnett square, which is our tool that we will use to obtain the ratios of the genome and phenotype of the first generation. How do you fill the Punnett square? Well, it's easy. The first column will be for one of the parents' alleles. In this case, I chose to place the alleles of the white organism. The first row is going to be for the alleles of the other. Later you will see that if you change these, the result will be exactly the same, but be sure not to mix the alleles of the parents yet. The mixture of the alleles will be inside the quadrants of the Punnett square. One rule to fill up the quadrants is to write first the dominant allele. That will make easier the study. So, in the first quadrant, we have a dominant allele that will be mixed with a recessive allele, and the result will be in this square. And then we go with this allele that mix with this one, and the result will go in here. Let's try to make it. If you can see, we have an uppercase letter from the first parent, a lowercase letter from the other one. This ratio of the offspring will be heterozygous. We have another uppercase, another lowercase, and if you see it, all the rest of the Punnett square will be the same. The problem asks for the ratios of pheno and genotype of the offspring. We have it inside the Punnett square. The genotypes for the offspring will be all heterozygous. Whether we know that it's heterozygous, remember that heterozygous means different genes or different alleles. It will be one uppercase and one lowercase. In this case, the offspring will be 100% heterozygous. What about the phenotypes? Remember that Mendel established a rule of dominance, which says that if the genotype has at least one dominant allele, that will be the one winning the contest for the phenotype, and that will be the trait that will be expressed. Because all of the offspring have at least one dominant uppercase allele for red 4, they will be 100% red. You have finished your first genetic problem using Punnett squares. Let's go to another example. We're going to use the same case, red 4 organism dominant with a white 4 organism recessive, but in this case, the genetic composition of the parents will be different. It says, if a cross is made between two heterozygous organisms, calculate the ratios and genotypes and phenotypes for the first generation. What is the new thing that you have here? The parents are going to be heterozygous. Hetero means different alleles. So again, you have in the problem the phenotypes and the genotypes of the parents. Because the problem says that they are heterozygous, we're going to put them like this, an uppercase and a lowercase for each of the parents. Remember the rule of dominance? If the organism has at least one dominant allele, that will be the one winning the phenotype. In this case, both organisms will be red 4. Now, let's go for the gametes. Remember the law of segregation. When the organisms produce their gametes, each gamete will have one of the possible alleles. One gamete will be dominant A, the other gamete will be recessive A, and the same will happen with the next. Having the gametes, the rest is very easy. You place them in the Punnett square. These are the gametes for the first parent. These are the gametes of the second one. And then we're going to start mixing them up inside the Punnett square. Remember, this allele joins with this one and goes in here. This allele from this parent joins with this one and goes in here, and the same with the rest of the table. So for the first combination, we can have in the offspring of this cross dominant homozygous, which means two uppercase. In the same row, we're going to have an upper and a lowercase, which means heterozygous. The same would happen in the next square, and in the last one, we will have two recessive alleles or recessive homozygous. Let's go with the genotypes. You already have them here. Remember that these squares inside the Punnett square represents the possibilities for having certain trait or certain genotype. The three first squares represent red 4 because remember the rule of dominance. If you have at least one dominant gene, that will be the one winning the phenotype. The three first squares represent red color. The last square represents white color. Why? Because we have two recessive genes. Remember that recessive genes need to be together to make the work or to have the phenotype. Now, because the problem is asking for the ratios, each square inside the Punnett square is going to have a 25% worth. Remember that the internal part of the Punnett square does not represent actual organisms. They represent the possibility of having certain genotype or phenotype. So it's going to have 25% multiplied times 4, the 100% possibility that we have for the offspring. So in the case of the phenotype, we'll have 3 squares with uppercase, 75% chances of being red 4, and just 1 square of having white 4, which is the 25%. The same will happen with the ratios of the genotypes, 25% being dominant homozygous, 2 uppercase, 50% being heterozygous, which means 1 upper and 1 lowercase, and 25% being recessive homozygous, 2 lowercase letters. Now, let's talk about the exemptions that we saw in a previous video. The case is going to be the same, but the problem says they are going to have a codominant condition. If the problem established that, then the things will be a little bit different. We're going to start with 2 heterozygous organisms, like that. But because of the codominant condition, remember we're going to have the phenotype shown at the same time, the phenotype of both alleles, we're going to have red and white 4 at the same time, which means spotted phenotype, a new one. Then we're going to have the gametes as always. We place them in the accurate position in the Punnett square. The genotypes of the offspring will be exactly the same as the previous exercise, but the phenotypes will change. The first square, dominant or uppercase letter, will be red 4. The middle section of the square will have an upper and a lowercase. They are both going to be spotted because of the codominant condition. And finally, the last square, 2 recessive alleles, will have white 4. That will change our ratios. This is the topic for the next video. I hope you have enjoyed the way to answer simple trait crosses. Remember to use your scientific thinking and to love biology. See you later. Bye.